drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, I'm going to take just a minute to tell you about Spores MD, your trusted source for premium mushroom products designed for research and educational purposes. At sporesmd.com, you can discover pharmaceutical-grade mushroom liquid cultures featuring dozens of unique strains of psilocybe mushroom species ideal for microscopy studies and mycology enthusiasts. They also offer culinary liquid cultures for gourmet mushroom enthusiasts, plus innovative options like nootropics gummies and Amanita muscaria tinctures, all produced in ISO 7 certified clean rooms with rigorous testing for viability, sterility, and purity. With a 100% satisfaction guarantee, Spores MD ensures high-quality products free from contaminants and without traces of controlled substances. Whether you're interested in scientific exploration or simply curious about the fascinating world of fungi, Spores MD provides reliable tools to support your interests, always in compliance with applicable laws. Head to sporesmd.com today and use the discount code TRICHOMES, TRIKOMES, for 20% off your purchase today. So go to sporesmd.com and check out what they have. So thank you for joining me.
Thank you again for the invitation.
Do you want to start off by just telling everyone a little bit about who you are and what you study?
So I'm Javier Gonzalez-Maeso. I'm professor in pharmacology and toxicology at the Virginia Commonwealth University. As a background, I'm originally from Spain, from a small town in the north of Spain. I did my bachelor there in biology, then my PhD there in the School of Medicine, doing research on receptors, G proteins, how the receptor is able to activate G proteins. And I was lucky to have access there in the lab to human brain samples. So we had, I think, interesting findings in postmortem human brain from individuals with depression and also heroin addiction. And then I decided to do a postdoc. So I came here to the US quite a few years ago. And I went to Mount Sinai in New York. That was a big change. And I was lucky to be involved in a lab working with psychedelics. So basically, the reason why I chose to join that lab as a postdoc was that they were proposing to use psychedelics back then in the early 2000s to study, again, back then, a new concept in molecular pharmacology, which is what we now know as the biased agonism. Back then was called agonist trafficking, which is how different drugs, chemically, structurally, very similar, activating the same receptor, the same molecule, are able to modulate or activate different pathways.
I see. So biased agonism. So this is the concept that one drug can activate a receptor, and another drug can activate the same receptor, but the internal pathways inside of the cell that those receptors are hooked up to, there can be multiple pathways. And so one drug can activate one pathway more through this receptor, and the other drug might activate another pathway more through this receptor. So same receptor, two different drugs, but you can get two different effects because of this concept of biased activation.
Biased agonism. And again, back then was a pretty novel concept in molecular pharmacology, and we were proposing to use to compare the effects of two drugs that you may know. One of them is LSD, classical psychedelic, and the other one is a lisuride. Lisuride is chemically very similar to LSD, but as you know, LSD is a psychedelic, lisuride is not, and is clinically used to treat, for example, Parkinson's. Again, back to your question, that was how I started to work with psychedelics. And from there, faculty, and now moved to ECU and working in this field, gene protein gamma receptors, behavioral pharmacology, molecular pharmacology, and psychedelics.
Yeah. So you've been working with psychedelics since the early 2000s. So quite a while before it kind of exploded into the hot field that it is now.
Yes.
Yeah.
And back then, when I was, for example, attending the SFM meeting, one of these meetings, many people told me, why do you work with psychedelics? They are not so interesting. And I said, that I think an interesting pharmacological tool. And now everyone is working, they want to work with psychedelics. So I'm very happy that we were able to convince them that psychedelics are interesting.
Yeah. So there's a lot we can talk about, and we're going to talk about some of the recent work that you've done. But you've already sort of alluded to the fact that so there's many different drugs out there, and there's many different drug classes. Psychedelics are one class we can talk about, but even within that single class, we can talk about one receptor, like the serotonin 2A receptor. And even there, you can have two drugs activating the same receptor, but they can activate it in different ways. So they can be very different in their effects, not because they're activating different receptors, but because they're actually activating or interacting with the same receptor in a different way. You mentioned, LSD activates the 5-HT2A receptor and it's psychedelic. You can have a very similar compound that's not that different from LSD, and it activates the same receptor in a different way, and it basically has little to no psychedelic effect. Another concept I think we'll talk about today is the concept of isomers and enantiomers. So what does that mean, just for people who don't know? What is an enantiomer or an isomer, and why is that important to know about if you're interested in drug action in the brain?
That is one of the interesting things in medicinal chemistry and pharmacology. How is the same drug that in one particular atom, the location is different, like a mirror image. Is, for example, the hands. So these could be isomers. They are the same, but one is the mirror image of the other. So if we move to molecules, to organic compounds, that is what we understand as stereochemistry. And again, we've done some work with that, only collaboration with faculty in the School of Pharmacy here at ECU. Richard Lennon, he's also been working with psychedelics longer than me, and Marcos Educat, again, they have a very strong program in medicinal chemistry, and collaborating with them, we are trying to understand how these isomers can induce different effects. And the most recent work that we did, it wasn't with classical psychedelic, it was with MDMA.
So enantiomers are one type of isomer, and the analogy here for people to think about is gloves. So if you have like winter gloves on your hands, you've got a left hand and a right hand. Your hands are like structurally the same. Your fingers are attached to your hand in the same configuration, left hand and right hand, but they're mirror images of each other. So if I try to put on my right hand glove onto my left hand, it doesn't quite fit, even though it's the same object in terms of spatial arrangement.
And the same will happen if you look into that. You mentioned you send the glove and the hand. Let's say the glove is the receptor and the hand is the ligand. So again, it's like if you want to use a ligand that is a mirror image of the other one and doesn't fit into the glove between commas of the receptor.
So many drugs have two versions of each other that are mirror images of one another, analogous to a left hand and a right hand.
LSD is another example. There are four isomers of LSD, and we think that only one is psychedelic. The other three are not. So again, that is another example.
Oh wow, yeah. So Okay, so LSD is a really complicated molecule in terms of its structure. There's actually 4 isomers. So it's the same arrangement of atoms. They're just sort of positioned slightly differently with respect to like a plane of focus. And LSD is psychedelic, but we don't think the other ones are at all.
In principle, no. Yeah.
Wow. And so another example of this that you've looked into recently is the example of MDMA. So like many compounds, there are 2 enantiomers, a left-handed version and a right-handed version. Those are abbreviated to S and R, correct? So that refers to each of the 2 isomers?
The position of that particular atom, yeah.
Yeah. So in terms of MDMA, let's assume that people know the basics. I've talked about MDMA quite a bit on the podcast, but with respect to the 2 enantiomers, the 2 isomers or the 2 versions of MDMA that are out there, S-MDMA and R-MDMA, before your recent study, what did we know about these 2 things other than that they existed? Do we know anything about any functional differences between them?
In human studies, clinical studies, there's not too much work. There's a recent study that they tested both, and they didn't see much differences. I mean, we mentioned that in one of our recent publications. In animal work, mice and rats, there is quite a lot, and there are many differences. Of course, in behavioral pharmacology, different labs have different findings. But again, it was kind of accepted that yes, comparing. Let me go back a little bit. MDMA is like a mixture, a chemical mixture, if I can say that, structurally speaking, between a mescaline, a classical psychedelic, and a methamphetamine. So I mean, it's like MDMA could do something similar to what both mescaline and methamphetamine do. And
I see. So MDMA is methylene dioxymethamphetamine. You're basically saying, from a structural standpoint, it's sort of somewhere in between methamphetamine and mescaline.
Mescaline, yeah. And again, there were several studies in the 90s, even 80s, testing the behavioral pharmacology of MDMA, and back to the question about the isomers, and some were proposing that the S was more amphetamine-like and the R was more mescaline-like, but depending on the behavioral model, others proposed the opposite. Again, the question was still open. I mean, and again, most importantly, I mean, that was before we kind of were convinced, if I can say that psychedelics could be used therapeutically. And now, I mean, you know this, I mean, one of the phenotypes that we think predict or suggest that the drug could be used therapeutically is their effects on structural plasticity in the cortex. So that is something that we tested, you know, how these 2 isomers, the R and the S, affect plasticity in mice, and we saw interesting differences between both.
Got it. So we've got these 2 versions of MDMA, S and R, the left-handed version and the right-handed version, but we'll just call them S-MDMA and R-MDMA. Many drugs have these 2 versions. This is very common. Ketamine is like this. You know, LSD, you mentioned, has 4 different versions because it's an even more complex structure. So you've got 2 versions of MDMA. The question is, do they do different things? Do they have different behavioral effects in animals? Do they feel different when people take them? Do they have different effects on plasticity, and so on and so forth? There was some hints in the literature that perhaps S and R-MDMA had different effects in animals, but the results were a little ambiguous or conflicting. But a priori, like without knowing anything, is it common for different isomers to have different effects? Like would we expect them to be pretty close to identical in their effects, or would we expect them to have some kind of difference in their effects?
Usually they behave differently. I mean, both molecularly in tissue culture, in cells, and also in animal models. They usually induce different effects behaviorally and in terms of signaling, cell signaling.
And so the other piece of background here that I want to discuss before going into this paper is, okay, so on the one hand, we've got the fact that drugs can have isomers or different versions of each other. There's S-MDMA and R-MDMA. On the other hand is the concept of sex-specific drug effects. So males and females may or may not respond the same way to the same drug or the same isomer of one drug. Sure. Can you just talk about that a little bit? How common is it for there to be sex-specific effects for drugs in terms of their behavioral effects and their pharmacological effects? Is it more often than not true that males and females respond differently, or at least not quite identically?
In the past, that's a very good question. And in the past, let's say 5 years or so, we've had many findings on in mice showing very, I think, interesting and intriguing sex differences between male and female mice. Let me tell you something about the story. So before that, most of the labs we used only males because for reasons that basically, I don't know, but we used males. And one of the students, PhD students, Elena Yaster, so she told me that why do we don't test to compare experiment design to test to compare the effects, not MDMA. Back then was psilocybin, psychedelic, and DOI on behavior in male and female mice. So we tested that. It was a cohort. They were matched of male and females, the same age, all little mates, and intriguingly, the females in the behavior that we tested, one of the behaviors, head twitch, we can talk about this further, but with that behavior, with the same dose of psilocybin and also DOI, another psychedelic, the females responded more.
Responded more.
Yes, responded more.
And that would imply, so the head twitch response in a mouse is used as a proxy for the hallucinogenic or psychedelic effects. That would imply that in your guys' hands, psilocybin and DOI were causing a stronger psychedelic effect in females at the same dose compared to males.
Yes. But, for example, again, that is for the trip, if I can say that. For the again, it's a model. We know that mice have many limitations, but that is, as you said, the behavior, the proxy that we use. We know that there's a nice correlation between the head twitch and the psychedelic effects in humans. So, and we also know that's important that the head twitch, it is principally serotonin dependent. So we know that those effects are serotonin dependent. But again, that was when we were testing the psychedelic or the hallucinogenic effects of these drugs. But when we were testing the therapeutic effects, for example, measuring reduction in models of opioid addiction, or measuring the effects of psychedelics or the effects of MDMA on dendritic spines, in most of the phenotypes, we don't see these effects in females at the same doses. So, I mean, again, those are the data. I think that it is very clear that there's a difference, at least in mice. The mice acutely modeling head twitch, modeling the psychedelic effects respond more, the females with the same doses. But when we test the effects, what we call post-acutely, 1 day at least after the psychedelic has been given, has been administered, so the drug is fully metabolized, but we see behavioral changes that we use as a model of clinically relevant effects. For most of the phenotypes, we don't see those effects in females at the doses. So, I mean, that is very intriguing.
Yeah. So I guess, the point here basically is that you guys have given drugs such as psilocybin and DOI to both males and females, same drugs, same dose, and they see similar effects. They have a effect behaviorally. They have some effect on plasticity. They have some post-acute effects on behavior afterwards, but the magnitude of the effect is different. So males and females are not responding identically, at least the rodents in your labs, to those psychedelics, which would imply that perhaps for something like MDMA, you might see distinct effects for males and females as well.
Yep. Yes, I mean, going back to what you said, we were talking about first with MDMA. So again, we tested 3 phenotypes, 3 readouts. We tested head twitch, this model that we mentioned. We tested one that is, we know it is downstream in terms of signaling of the serotonin 2 receptor, accumulation of AP1, one of the pathways downstream. And we also tested dendritic spine. So, I mean, we and others have shown, and you may know this, that psychedelics, also ketamine, also traditional antidepressant, but that is a different mechanism. But psychedelics increase these, what we know as dendritic spines, these small protrusions in the dendrites, and we think that is one of the phenotypes responsible for the beneficial, for example, antidepressant effects of these drugs. So again, with the different isomers, DR, MDMA, and S-MDMA, there were sex differences and also differences between the two isomers.
Yeah. So basically, in the lab, you've sort of got 3 types of readouts you can use when you're studying the mice in terms of their responses to these drugs. You can look at their behavior. What are they doing when they're moving around and behaving and doing things? Two, you can look at structural plasticity, like dendritic spines. Are more spines growing? Are they going away? You can see physically how neurons are changing in response to them taking these drugs. And the third would be intracellular signaling. So stuff happening inside of the cells, different information pathways can go up or go down and things like that. So you can look at the neurons and see how they're physically changing. You can look inside the neurons to see how changes are happening inside of them. Then you can look at the whole animal to see how it's behaving. And in general, for drugs like psilocybin and some others that you mentioned, you see differences between males and females.
Yes.
Yep.
Yeah.
And so going back to this concept of enantiomers or isomers, there's two different versions of MDMA, R and S. There was some indication in the literature previously that they could have somewhat distinct effects, not completely distinct, but somewhat distinct effects. I think the idea was that the S-MDMA isomer is more amphetamine-like. Maybe it has more effects on locomotion behavior and feeling, the amphetamine style effects. And the R-MDMA isomer was perhaps thought to have more of the entactogen or empathogen effects, so the effects on social behavior and sort of the what we like to think of as the lovey-dovey feeling that the mice would get, similar to what humans experience on MDMA. And so you did a paper recently where you actually tested some of these things, and you looked what happened in males, what happened in females for R-MDMA and S-MDMA. So can you walk us through some of the results a little bit? What did you start to see there?
So again, I think one of the most intriguing findings is that for the two isomers were able to, what we call in pharmacology, bind the serotonin to a receptor. But when we measure function activity of the serotonin to a receptor, only the S showed, or in cells in tissue culture, a very weak effect activating the serotonin to a receptor.
Got it.
The R did not.
Got it.
But when we went into the head twitch, so the effect of the S was much stronger than the effect of the R. And there was also a sex difference. And when we measure spines, I think is probably the most intriguing finding of this paper is that only the S at the same dose. We used a dose that induced head twitch in male and females, the same dose when we tested dendritic spines. And again, I mean, the mice received single dose of the two isomers, R and S, and the samples were collected 1 day after, once the drug is fully metabolized. So only the S and not the R MDMA was able to increase these dendritic spines. And not only that happened in male mice, but not in female.
Interesting. So backing up for a second. So in the classic psychedelics like LSD and psilocybin, you see this head twitch response in mice, which is taken as a proxy for their hallucinogenic effects. So drugs like psychedelics like psilocybin and LSD, they bind to the serotonin 2A receptor strongly. And we know that that's necessary for their hallucinogenic effects. We know it's also necessary for this head twitch response in mice. So literally the mice will kind of twitch their heads like that. And we like to think that this means they're probably having some kind of hallucinogenic effect as well. So classic psychedelics bind to a receptor, they induce the head twitch. You guys measured 5-HT2A receptor binding and the head twitch in mice to S and R-MDMA. Basically, what you found is that only the S isomer of MDMA binds to 2A, not the R isomer, but it does so pretty weakly, much more weakly than a classic psychedelic.
So what we think, I mean, we went into some using pharmacology to study how that happens. And what we think is that, again, that's still an open hypothesis, an open question, but we think that this head twitch and also what the S isomer of MDMA induced on the spines is not because of the agonistic effect at the 2A directly, but because, I mean, we know others have shown that MDMA induces a huge release in serotonin.
Yeah. So you think it's an indirect effect. It's not like LSD, which directly binds the receptor strongly. MDMA is weakly or not even binding the receptor at all, but it's inducing the binding of serotonin to the 2A receptor because it's causing a huge increase in serotonin.
Serotonin, I mean, mechanistically, we know this MDMA gets into the presynaptic terminal through the serotonin transporter, is inducing a huge release of serotonin to the cytoplasm, and it goes back out to the synapse. So it's like a huge peak of serotonin. So we know that, when we did this, we gave mice fluoxetine, which is a blocker of the serotonin transporter.
Yeah.
And we saw that with fluoxetine, we are able to block the head twitch induced by the S isomer of MDMA, but not the head twitch induced by DOI, which is a classical psychedelic.
Got it. So without even blocking the 2A receptor, you can block the head twitch of the S isomer because you're blocking its ability to go through the serotonin transporter.
And induce this huge, in principle, that huge release of serotonin.
Yeah.
It's activating the 2A, MDMA is activating the 2A, but not as classical psychedelics do, binding the orthosteric binding pocket of the 2A. But we think that is probably because of this huge increase in serotonin in the synapse, and that serotonin is activating the 2A.
Mm-hmm. I want to make sure people understand this piece, the mechanism of action here. I think a lot of people understand that MDMA affects serotonin. It leads to an increase in serotonin. But I want to walk through this mechanism one more time just so people get it. So you're saying that the way that MDMA works is basically that it goes into the presynaptic terminal. So it's going inside of a neuron that's going to release neurotransmitters.
There's much more work to do there. We think that is one of the hypotheses. Yes.
Yeah. Okay. But anyways, walk us through this again. So what is the serotonin transporter? What is its normal function?
Serotonin transporter to get back the serotonin from the synapse to the presynaptic terminal.
Got it. So like a neuron would be releasing serotonin, a serotonin neuron, and there's a transporter called SERT, serotonin transporter, and it's basically for recycling synaptic serotonin. So the cell will release a bunch of serotonin, and then it will want to recover that and reuse it again, and it uses the serotonin transporter to do that.
The traditional antidepressants. One of the mechanisms is that is to block the serotonin transporter, so the amount of serotonin in the synapse is supposed to be increased to keep higher the concentration of serotonin in the synapse.
Got it. And does MDMA leads to an increase in synaptic serotonin via the serotonin transporter. Does it affect other neurotransmitters as well?
Yes, I didn't mention that yet, but yes, it is not at all specific to serotonin. It targets also the dopamine and the norepinephrine transporter. That is a different question, a different thing. What happens via those mechanisms, we haven't tested that yet. Dopamine, norepinephrine, those are supposed to affect behavior, plasticity, you know that, but we don't know what MDMA is doing on those transporters yet.
Got it. So the mechanistic details are still mysterious, but we do know that MDMA increases not just serotonin, but also norepinephrine and dopamine as well.
Yeah.
Interesting. So you found divergent effects in males and females for MDMA. The S and the R were different from each other, but there was also a difference in structural plasticity between males and females. So remind us again what you found there. You're looking at dendritic spines and how many spines are popping up or going away in males and females. What did you see there?
So we found that with the S isomer, we saw an increase in the spine, the density of spines in the frontal cortex. That didn't happen with the R isomer, and what we saw in males with the S isomer, it didn't happen in the females.
Got it. And so the result here, an increase in dendritic spines, this is something that people have seen in various model systems with various drugs, like psilocybin, like LSD, like ketamine. And it is thought that this rapid increase in dendritic spines, this increase in structural plasticity, might underlie some of the therapeutic benefits that people have seen with these drugs. What you're saying is you saw an increase in dendritic spines in the frontal cortex with S-MDMA only, not R-MDMA, but only or mostly in males, not females.
Yes. Yep.
Interesting. And so that would imply that there could be therapeutic effects that you would see only with S-MDMA and only in males, but not with R-MDMA or in females, even with S-MDMA.
Yeah. And again, this is, I think, interesting. We've shown, we may talk about that also. We've seen differences also with psilocybin in the effects of this drug on models of opioid addiction. And again, it is also differences between male and females. And when I discussed this with researchers doing clinical work, they say that they don't see too many differences between male and females people. Why? I don't know. But again, one of the reasons why is because, again, I'm not a clinician, I do basic research, but my impression is that they don't include or they don't have enough statistical power to X as an experimental
Yeah.
as an independent experimental variable, and also they usually administer or give the same dose to male and females, and usually in the clinical studies they do not correct the dose with the weight of the individual.
I see. So they give everyone, say, X number of milligrams, even if you weigh 200 pounds or 120 pounds. And isn't it also true? My understanding is that in clinical settings, in human clinical trials that have used MDMA, they're mainly, or I think even exclusively so far, using a racemic mixture of both the S and the R isomer rather than choosing one or the other. Is that true?
For MDMA, yes. I mean, there's a recent paper that they tested the R and the S, but before, yes, they used the racemic mixture. Yeah.
Got it. So most of the clinical studies that have been done already that people may have heard of out there, like the MAPS trial for MDMA-assisted psychotherapy, that was done in both males and females, but there was one dose given to everyone that may not have corrected for weight, and it was a mixture of the S and the R version. It wasn't one or the other.
Yep.
Got it. So basically, if you put all of that together, what your results imply is that if you were to start doing larger studies and you had more males and females and you used the S and or the R isomer separately, you might start to see differences in the therapeutic benefit, the types of things you see, in the magnitude of the effects between the two isomers, between the sexes.
Between the sexes, yeah.
Interesting. What else did you find in your MDMA paper with respect to the S versus the R isomer and sex-specific differences? Was there anything else there?
I mean, the way they activate the pathway downstream the serotonin. So again, it is a very weak effect. It was induced in both male and females, but it was only induced by the S, not by the R.
Got it. So in general, whether you're looking at structural plasticity or some of these intracellular effects on pathways inside of cells, the S isomer tends to have larger effects than the R isomer.
Targeting the serotonin 2A, yes. And that also correlates, I mean, in classical pharmacology, usually, if the compounds are structurally similar, in pharmacology, you know this, an agonist is a compound that binds the receptor and activates the receptor. An antagonist is a ligand that binds the receptor. It's not supposed to affect the receptor functionally, but it prevents the effect of the agonist. And usually, if the compounds are chemically similar, structurally similar, an agonist shows a lower affinity targeting the same receptor compared to an antagonist. And when we tested that, we used a classical assay for the serotonin 2A, displacing ketanserin, an antagonist for the serotonin 2A, and the affinity of the S was lower compared to the affinity of the R. So that also correlates with what we saw in vitro, in tissue culture, that the S saw the weak agonism and the R did not.
Yeah. So another question that comes up here is, when we think about people out in the real world using MDMA recreationally, and they're buying street drugs and things like this, if they're buying ecstasy tablets or MDMA in some form, is it typically going to be a mixture of the S and the R? And
I would say yes. Racemic, yeah.
I see. So people who are using it recreationally out there, they might report different effects, in two different times or two different substances that they encounter. So it's possible that sometimes when people are using MDMA, it's an equal mixture of the S and the R isomer. Sometimes it might be mostly one, sometimes it might be mostly the other, and that could account for maybe a variability in the effects that people experience.
I mean, in medicinal chemistry, racemic is supposed to be a 50-50 mixture, but in the street, I don't know.
You never know. But that's part of the point here, right? Is it could be that the mixtures could vary quite a bit in their proportions of S and R out in the real world, and so people never know exactly which combination they're getting.
Yeah.
Interesting. You've also done work on psilocybin and sex-specific effects that you've alluded to. You've looked at things like how psilocybin affects opioid addiction and reward processing. Yep. I want to give people a little bit of background here before we go into your paper. When you're talking about animal research, especially the types of research that you do, how do you guys study opioid use and addiction? How do you normally administer these things to the animals? And what does sort of the addiction and behavioral readout of that look like?
That is a big question. And maybe, frankly, I don't consider myself an expert in the pharmacology of drugs of abuse. I'm lucky that colleagues here at VCU They have labs working on that. So basically, yes, but back to the question, I mean, we are trying to model both the rewarding effects of opioids and also the effects of these drugs on negative effects, such as, for example, withdrawal. You know that after repeated administration of opioids to patients, after the drug, I mean, the individual start to see, for example, withdrawal symptoms, muscle dystonia, vomiting, deficits in the gastrointestinal motility. So, I mean, that is one of the reasons why individuals with opioid use disorder go back to the drug, because to reduce those withdrawal symptoms. So again, in the mice, in models, we try to model those behaviors differently, modeling rewarding aspects of in principle opioids that are supposed to be addictive, and also the withdrawal effects.
Got it.
Those are different phenotypes that we can test in mice.
Got it. So with opioids, you've kind of got two sides to the addiction question. On the one hand, the effects acutely can be rewarding. People like them. It feels good. There's euphoria. That's one reason why people go back to the drug again and again. But on the other side of the equation, we've got negative withdrawal symptoms. You've got gastric motility problems that cause gut issues. You've just got the negative effects and feelings that people get when they are no longer high on the substance and getting its acute effects, and they're feeling bad. And then they'll go back to the drug to relieve those negative symptoms. So sort of two sides to the addiction equation here with opioids. And you're saying you can model all of those things in rodents.
So, I mean, we started with that because, I mean, the same student, Elena Jaster, that convinced me to test these sex differences on the gut twitch. So when she joined the PhD program here at PCU a few years ago, so she came to the lab to propose us to start to model the effects of psychedelics, psilocybin, on these models of opioid addiction, opioid abuse. So we tested, I mean, a few more, but mainly two behaviors. One of them is what is known as, in pharmacology, is known as a condition-place preference. So this basically, you let me move this to the street. I mean, for example, in individuals with alcohol use disorder, when they go back, I mean, they don't wanna go back to a pub because just the environment is going to increase the willingness of consume alcohol, right? Because of the place, because of the space. So moving that to mice, to models. So basically, the mice received, in this case, we did it with oxycodone, an opioid, analgesic, but also with potential abuse liability. So the mice received in one chamber oxycodone 1 day, and the day after received vehicle, and the other way around. And after a few days, again, we associated one of the places in the chamber with either oxycodone or vehicle. We let them choose free whether the place, whether they had received oxycodone or vehicle. So that is what is called condition-place preference. And as many others have shown already, so yes, mice showed condition-place preference. So the mice showed preference for the place where they had received oxycodone the days before.
Got it.
That was in the morning. So again, nothing new. The mice showed preference to the place where they had received oxycodone. And we tested that in the morning. And then in the afternoon, the mice received one single dose of administration of psilocybin or vehicle. Go back to the home cage. Once again, the drug is fully metabolized. There's no psilocybin. And then the day after, in the morning, the mice went back to test for preference. And then, as again, you want to go back looking for the place where you had received oxycodone or no.
Yeah.
Again, that is, I think, very interesting finding. Mice, again, all of them conditioned with oxycodone. So they showed preference for oxycodone.
Yeah. So basically, it's like you've got two cages, cage A, cage B. You give oxycodone to mice in one of them, but not the other one. Yeah. And mice prefer the cage where they get the oxycodone. There's a rewarding effect there. They like it. If you give them the choice between two cages, they're going to spend more time in the one where they got the reward, the drug reward, in this case, an opioid. So by analogy, this would be like if you had two apartments that people could go into, and one of them they got to do their favorite drug, like an opioid, and the other one they didn't, they would tend to spend more time in the one where they got the drug reward than the other apartment. And so that's sort of your way to assess the rewarding effects of the drug in the mice. And then the experiment is you give them psilocybin and you see if that preference is maintained or it starts to diminish.
So what we found is that, again, mice that were showing preference to oxycodone, one day after that single administration of psilocybin, they saw that a decrease, in the preference for oxycodone. So they spend similar time compared to the chamber when they received vehicle. So a single dose, single administration of psilocybin reduced preference to oxycodone. It's like a traditional model of rewarding effects of the drugs.
Wow.
But once again, the dose of that's important. The dose of psilocybin that we used induced more head twitch, the hallucinogenic effect, in females than in males. It was the same dose, 1 milligram per kilogram. And that dose, even when induced more head twitch in females than in males, when we tested these effects on preference for oxycodone, the dose reduced preference for oxycodone in male mice, but not in females.
Wow. Okay, so there's a lot there. So you reduce this preference. So the mice, you give them oxycodone in one cage, not the other cage. They develop a preference for the cage where they get the oxycodone. That makes perfect sense. You're then giving them a single dose of psilocybin, 1 milligram per kilogram, both males and females. It induces a head twitch in both males and females, but more so in females. That implies that it's having a hallucinogenic or psychedelic effect in both sexes, but perhaps more strongly in females.
That implies, to basic pharmacology, that implies that the dose that we use is pharmacologically active in both males.
Yes. So it's pharmacologically active dose in both males and females. But then the place preference goes away more in the males than the females.
Yep.
So it's sort of like the interpretation here would be that the males and the females both develop an oxycodone place preference for one cage where they get the oxycodone drug. You then give them psilocybin, and that preference goes away or greatly diminishes in the males, but not so much for the females.
Not in females. I mean, it was basically testing. We had the 4 conditions for 2 drugs, 4 conditions: vehicle, oxycodone vehicle, psilocybin vehicle, oxycodone psilocybin. So the preference the day after psilocybin or vehicle between the oxycodone vehicle and the oxycodone psilocybin groups were the same in females, but in males, the preference in the oxycodone vehicle was much more than the preference in the oxycodone psilocybin groups in males.
So correct me if I'm wrong. These results would imply that in rodents, females are more sensitive to the psychedelic effects of psilocybin, but they're less sensitive to the extinction of place preference or drug reward memories.
Yeah.
Interesting. And so what does that sort of tell you overall in terms of what we should start to think about from a clinical perspective when we're thinking about trying to use or study things like psilocybin for addiction in human beings?
I mean, that is one, again, I mean, I'm very concerned about the limitations of the experiments in mind. Is what we do, is the tools that we use, but but it is not humans. So I mean, again, my message to the clinical researchers is that I think that we need more information about sex differences between male and females, about just the acute effects of psychedelics, enough statistical power, and if they are able to replicate some of these findings, basically sex differences in humans, so we can go back to mice and design the experiments in a better way, so we can understand what happens. Again, they are not much with the classical psychedelics or with MDMA. I think there's a preprint testing the effects of ketamine, and I think they are starting to see sex differences with ketamine.
Yeah. Again, so, you've got this result with psilocybin in this opioid addiction paradigm in mice. You see a sex difference. You looked at the R and the S isomers of MDMA and the behavioral and cellular effects of MDMA. You saw a sex difference there as well as an isomer specific difference. based on your studies that we just talked about and all of the other literature out there, I guess one of the take-home points here is that we should expect more often than not the drugs are going to have sex-specific effects, and they're not going to behave identically for males and females. And the other thing to emphasize is that in your studies in mice, you are tailoring the dose to the weight of the animal. So you're giving a 1 milligram per kilogram of body weight and things like that.
We selected that dose based on the behavioral effects on the naked twitch.
Yeah. So the sex differences are not just due to differences in weight. They're actually due to the difference between males and females.
Yeah.
Interesting. The other thing I want to ask you about is when we think about classic serotonergic psychedelics like psilocybin, DMT, LSD, when we start to think about the mechanism of action there with respect to their hallucinogenic or psychedelic effects, people often talk about the 5-HT2A receptor because that's the so-called psychedelic receptor. That's the one necessary for the hallucinogenic effects of these drugs. If you block it, those effects largely go away. And people often sort of just stop there. You take one of these drugs, it binds to the 5-HT2A receptor, and then psychedelic effects happen. But of course, that can't be all that's going on. There's a lot happening between cells, inside of cells, at different levels of analysis. You've also done some interesting work building on the mechanistic detail of how classic psychedelics act. So they bind to the 5-HT2A receptor, they affect it in different ways, and then other stuff is probably happening too. When a classic psychedelic is having psychedelic effects, what's going on at the cellular level in terms of mechanism beyond the serotonin 2A receptor?
That's a very important question. I mean, I think that, yes, when you talk about psychedelics, kind of by definition, it's like the effects are serotonin 2A dependent. I mean, we think that, yes, the serotonin 2A is an important piece in all the effects of psychedelics. But when I tell the students, I mean, it's not a correct term, pharmacological speaking. I always tell them drugs are usually very dirty. I mean, by dirty, I mean they bind, they activate, they touch many receptors. And the pharmacological profiles of all psychedelics and almost all the neuropsychoactive drugs, they bind a big battery of receptors. For the psychedelic properties of these drugs, we know that the 2A is necessary. I mean, there's a nice correlation, and actually the very first correlation in the early 80s between a potency of psychedelics activating the serotonin 2. In the 80s, they didn't have 2 and psychoactive effects came from BCU, Richard Lennon, and there was nice correlation. But again, now we know that there are other receptors involved. We know that the 2C is also an important target, the serotonin 1A, all the psychedelics activate also the serotonin 1A. Not much, for example, DMT or psilocybin, but LSD also targets the dopamine receptors. So I think that they are all involved. And going back to what you said, I mean, in mice, you know this, I mean, we have tools to delete the genes. So we and others have shown that for these effects of psychedelics in some of the phenotypes, behavior, and also in dendritic spines, gene expression, some other phenotypes, in the knockouts for the serotonin 2A, most of those effects are abolished. But there are also effects that are not serotonin 2 dependent. Let me go back to what we mentioned about spines. So we saw that with the DOI, it's not in the clinic, but is used in the labs, is very specific for the serotonin 2A. So DOI also increased density in the spines, and those effects are serotonin 2 dependent. With the psilocybin, we have some data still unpublished, but it's the same. So the effect of psilocybin on the dendritic spines in the cortex is also serotonin 2 dependent. But if you go to subcortical regions involved rewarding processes addiction, so a particular brain region is what is known the nucleus accumbens, the ventral part of the striatum.
Yeah, a key part in the reward processing circuit of the brain.
Yeah. So what we showed, and that is published already, we showed that is interesting. The same dose of psilocybin induced opposite effects in terms of dendritic spine density in male and females, increase and decrease.
Wow. So not just different effects, quantitatively opposite direction.
Opposite, increase and decrease. Not only that, those effects opposite to what these drugs do in the frontal cortex, in the subcortical region involved in rewarding processes and addiction. So those effects were identical in the knockouts for the serotonin 2A. So it seems like this drug, psilocybin for now, the effects, structurally speaking, in the cortex is serotonin 2A dependent, but in the accumbens, in the nucleus accumbens, even when it does, I think it's in drinking opposite things in male and females, those effects are not serotonin 2A dependent. At least mice using this genetic tool. So again, back to your question, there are probably other receptors involved in the effects of these drugs, and what these receptors do, we don't know. And
right. And if sort of people step back for a sec, a lot of this should be pretty unsurprising, I think. We talk about the serotonin 2A receptor a lot because it's famous for being necessary for the action of classic psychedelics, but all of the classic psychedelics are non-identical in terms of their effects. DMT will give you different hallucinogenic effects than LSD and psilocybin, and so on and so forth. And we know that all of these drugs will have a pattern of receptor interaction, and there's a lot of overlap between them, especially with some of the serotonin receptors, but they all have a distinct receptor pharmacology profile. DMT will interact with different receptors in a different pattern than LSD or psilocybin and so forth.
So again, each drug is different, the pharmacology, the only common, a high affinity for the serotonin 2A, many others, but the pharmacological profile is different to all of them.
And then, so you've added onto this the complexity of, males versus females. There can be sex differences here. And then on top of that, you've just told us that in different parts of the brain, you can have not just sex differences, but some of the mechanisms that work in the frontal cortex might depend on the serotonin 2A receptor. And then the same drug in the same animal in a different part of the brain might be doing things that do not depend on that receptor, but depend on other receptors.
Again, I mentioned this briefly, but I think it's about sex differences. Another important phenotype, I mentioned this briefly. We talked about the CPP, rewarding, but I think one of the issues about opioids is withdrawal, the negative effect. So we also, in collaboration with Iman Tamas here at BCU in Pharmacology, so he has a very elegant model of withdrawal in mice. So it's like basically a mini pump for a few weeks of the, in this case, oxycodone or vehicle. After that, the pump is removed and the mice start to feel, they don't feel well. And you can see that. Jumping, motility, these things.
Got it. So you give them opioids and then you take them away and they're not having a good time. They look like they're having withdrawal symptoms.
So again, back to the same model that we use with the CPP, long-lasting administration of oxycodone, removal, and then just after the mini pump was removed, single administration of psilocybin or vehicle. And the day after, measuring withdrawal visually. The students did this, and as expected, oxycodone induced withdrawal symptoms in both male and females. Psilocybin reduced those withdrawal effects in male, once again, but not in female. So,
got it.
So it's not only, psilocybin is not only reducing rewarding effects, CPP in that case, it also reduces the withdrawal effects, the physical negative effects of in this case, oxycodone. And that, again, at that dose, happened in male but not in females. So,
got it. So psilocybin can affect sort of the positive and negative symptoms of opioid addiction. It can affect the preference for the place where you got their drug reward. It can also diminish the withdrawal symptoms, the negative symptoms that come from being off the drug after you've been on it, the mice, I mean. And this is the effects there are stronger, again, in males than females in mice.
But again, mechanistically, in terms of circuit, cell location, they are supposed to be very different. Parts of the body, parts of the brain, or parts of the peripheral that affect rewarding aspects of these drugs, and parts of the circuit that affect these physical withdrawal symptoms.
Right.
We know what we do. That psilocybin reduces that. It is supposed to be at least at this dose sex specific, but where in the body is this drug targeting, so that the same drug is able to reduce addiction and withdrawal? That is also an open question.
Got it. Got it. So we're still figuring out those details, but a lot of these pieces do seem to fit together, at least at a high level. You're seeing different behavioral outcomes in male and female mice at the same dose of the same drug, like psilocybin. And you're also seeing, you and others in the literature have seen, that the changes inside of the brain, such as structural plasticity, can be different in males and females, and even in the opposite direction, even in, or especially in places like the nucleus accumbens, which we know are critical for things like reward processing. So exactly the parts of the brain where you would expect to see differences, given what you've seen at the behavioral level.
Yep. Yep.
Interesting. So going back to sort of the mechanism of action and the way that classic psychedelics work beyond the serotonin 2A receptor, you've looked at something called the metabotropic glutamate receptor. Can you give talk about that a little bit, but let's give people the basics here. What is that receptor and what is it sort of known for in neuroscience generally?
Okay. So, I mean, there is a different, let me go back. The serotonin 2A, it is, it is what we know as a G protein kappa receptors. So they activate intracellular G proteins, receptor inhibitory. There's another receptor that is a glutamate. Most of the glutamate receptors are ion channels, but they are also GPCRs, G protein kappa receptors. And there's one that is expressed in similar cell types as the serotonin 2A that is known as the metabotropic glutamate 2, metabotropic glutamate receptor 2. This one is supposed to be inhibitory in terms of signaling, canonical pathways. The serotonin 2A is supposed to activate G proteins that are excitatory, GQ G proteins that induce calcium release and those pathways. The metabotropic glutamate 2, or NGLUR2, is a GPCR, but it's supposed to be inhibitory. It recruits GI-dependent signaling, and that reduces the production of cyclic AMP and those pathways. So many labs, including our lab, have shown that there's a functional interaction between these two receptors. Activation of the NGLUR2 is able to reduce the pathways induced by the serotonin 2A. And we found that these two receptors can interact physically, what we know in biochemistry as a dimer, so two receptors together, and one of them can affect the function of the other. So what was intriguing, for example, one of the most intriguing findings, I think, with this NGLUR2 is that back to psychedelics. So we found that knockouts, genetic mouse models, knockouts for the NGLUR2, for the metabotropic glutamate 2. So these mice, they do express the serotonin 2A. So the molecular target of psychedelics is still there. But when we tested head twitch in these mice, the head twitch was almost absent. So that looks like the serotonin 2A by itself is not able to induce this behavior, the head twitch, without the NGLUR2. So that is, I think, intriguing. And in terms of coupling, we also showed, and actually there's a paper in Nature published last week from a group in China, a very strong group, and they replicate that. We saw that back to what we discussed briefly at the beginning, the difference between psychedelics and non-psychedelics. Psychedelics target the 2A, induce in mice head twitch, in humans hallucinations. Non-psychedelics do not. We proposed that one of the potential differences in terms of signaling between psychedelics and non-psychedelics is that psychedelics, for example, DOI, mescaline, LSD, psilocybin or psilocin, the active metabolite, targeting the serotonin 2A, are able to recruit both the canonical pathway, GI, and also the non-canonical pathway, GI, inhibitory. And non-psychedelics, for example, lisuride, ergotamine, some others, even when they target the same receptor, they were able to activate only GQ. But what we think is that going back to the NGLUR2, we think that at least, again, in vivo, in the cortex, that the serotonin 2A is able to activate both GQ and GI because it does it together with the NGLUR2.
Got it.
That is one of the hypotheses that we have, and that one of the hypotheses that we are testing.
Yeah. So basically, people can think about receptors in the brain as belonging to two large classes. On the one hand, there are ion channels. They physically open and close, like physical holes in the cell, pores, to allow sodium or calcium or potassium or all of the above to come in and out of the cell. But then you've got another type of receptor that's a metabotropic receptor. It is not an ion channel. It doesn't physically open with a pore. It is bound by a drug or a ligand. It might be a psychedelic like LSD. It might be a transmitter like serotonin. On the outside of the cell, it's bound by one of those things that changes what it's doing on the inside of the cell. And these non-ion channels, these metabotropic receptors, like the 5-HT2A receptor, like the metabotropic glutamate receptor, they can be hooked up to different protein signaling pathways on the inside of the cell. And they're often hooked up to more than one. They can be, pathway 1, pathway 2, pathway 3. And the drug that binds on the outside can mostly activate pathway 1 on the inside, or maybe pathway 2 on the inside, or maybe both pathway 1 and pathway 2. And so what you're saying is psychedelics activate the 5-HT2A receptor, and they've got two different pathways on the inside of the cell that they seem to tap into. And then on top of that, there's these metabotropic glutamate receptors, not serotonin receptors. They also seem to be involved at some level. And you can actually have dimers of different receptors. You can have sort of half of a 2A receptor for serotonin and half of a glutamate receptor come together to make a new sort of hybrid receptor. And those seem to be involved in psychedelics as well.
Yep.
Interesting. So what are some of the new questions and the upcoming work that you're doing that builds on some of the things that you've done already that we've discussed today?
To me, one of the, back to psychedelics and plasticity, to me, one of the open questions is to link causality between, is not an easy question, to link causality between those structural effects and behavior. For now, we have correlation. More spines, more memory, more spines, less depression-like behavior. But how do we know that that is necessary or sufficient for what we have in terms of behavior? So that is one of the questions. Back to the sex differences, I think we need to extend our knowledge in terms of pharmacology. More doses are also what happens, for example, in females. For now, we haven't done anything with the estrous cycle, the menstrual cycle. That could be something intriguing, and that's something that, if we model that in mice, we could maybe get that expanded into clinical studies. More generally speaking, and we didn't talk about this, and to me, again, I mentioned this, I'm not a clinician, but one of the, I'm not sure about the best word, one of the potential concerns about the studies in humans is the placebo effect.
The placebo effect.
Yes. In almost any clinical studies, the individual is not supposed to know if they receive the active molecule or a piece of sugar.
But with psychedelics, you can't really do that because if you're tripping, it's obvious.
Of course. And also the individuals that are willing to be exposed to the experience. Not everyone want to get under the effects of psychedelics. So I think there's kind of a bias in the population that is willing to receive that. And also, and I think that is an issue, the placebo effect. Again, I've been thinking about that. I don't think it is easy to design a clinical study in which the individual doesn't know if they receive psilocybin, MDMA, ketamine, or not. But I think, that is, again, we are trying to model that in mice, to model the placebo effect, to try to design models to understand that. But I think that is an open question in the field in general. How, again, in pharmacology, different fields, schizophrenia, cancer, we are trying to develop like personalized medicine. One patient, one particular profile of symptoms, one treatment. In this case, one drug, one psilocybin for depression, PTSD, anxiety, anorexia, pain. I didn't mention this, but we also have findings showing that psilocybin reduces pain in mouse models. How can that be? That one single molecule can be used to treat such a diverse group of diseases. That is like a magic bullet, right?
Right.
So it's happening. So, I mean, we need to understand better. And I'm not questioning, of course, it's mice. In mice, we don't have a placebo effect. We don't have like the predisposition to be exposed to.
Right.
And they have less pain, they have less addiction, they have less withdrawal, so it's working. So how can the same molecule, psilocybin, can be used in mice to treat such a variety of diseases? It's something, I think there's something, it must be something there that links together all these things. And I think that is one of the most intriguing questions that we have in the field.
Yeah. No, it's interesting. And I think, I've talked to people like Gul Dolan on the podcast before, and she's done experiments where, they do psilocybin and ketamine and MDMA. They look across drugs. And I think what she would say there, what she has theorized is that all of these drugs are converging at cellular mechanisms that are downstream of the receptors they interact with pharmacologically to have epigenetic changes that enable these circuits to become labile and plastic again so they can be changed. And that's her explanation for why you can see one drug, and actually multiple drugs, that can each be used and have efficacy for multiple disorders, because they're sort of enabling the cell to become changeable again for some time, no matter what the change needed actually is.
Yeah, basically, this concept of plasticity, to increase the plasticity of the cells, that is possible. I mean, what you said is right, totally right. Different drugs, different receptors, different powers, but they come on something at the end that brings together all those mechanisms of action.
Yeah. And so, among other things, I think some of the takeaways from what we discussed with respect to your work, Javier, are that, there are multiple versions of many drugs. You can have different isomers like R-MDMA and S-MDMA. A lot of drugs work this way: ketamine, LSD, a lot of drugs. And also there's sex-specific effects. So when we think about drugs, we have to take both of those things into account. Males and females will more often than not respond differently to the same one isomer of a drug, and they both might respond differently than that to the other isomer of a drug. And so for better or worse, it's really complicated, but we have to keep all of these things in mind when we're thinking about the effects of these things, how the results might generalize across the population, and who they might work for and not work for, because they're probably not going to have the same effect in everyone.
Yeah.
Are there any final thoughts you want to leave people with, or anything that you want to reiterate from our discussion today?
No, again, I try to say, the fact that we have, most recently MDMA. Again, I've been working with MDMA in the past few months. It's not, my main field. Psychedelics, quite a few years working with psychedelics. I think they are interesting. I'm very happy that, young investigators become interested in this field. And again, I think we have many, many open questions that happens always in science. And I think one of the two main questions are the pathways that distinguish psychedelics from non-psychedelics. If the trip, I didn't mention this, but if the trip by itself is necessary for the beneficial effects, we and many others are developing new non-psychedelics. So drugs that target the 2A and others do not induce head twitch, in principle do not induce hallucinations in humans, but in mice, they are therapeutically efficient. So the question is, if we move that to clinical studies, is the hallucinogenic experience part of the beneficial effects of these drugs? That is, I think, an interesting question.
Yeah. All right. Well, Dr. Javier Gonzalez-Maeso, thank you very much for your time. I appreciate it.
Thank you.
Hey everyone, a great way to support my efforts is to visit my support page at mindandmatter.substack.com. A link to that page is in the episode description, or you can search for it on the Substack page itself. The support page has an up-to-date list of my affiliate partners. These are companies I work with, and if you buy those products through those links or using the discount codes provided, those will get you a great deal on a variety of products related to optimizing physical, metabolic, or mental health and vitality. These are all products I use myself, and some of them are directly related to podcast episodes or formulated by Mind and Matter guests. For example, there's a great product called Keto Citra, formulated by a kidney biologist who is on episode number 186. It contains the ketone body BHB together with several minerals in a precise ratio formulated with kidney health in mind. There are also links to products like physical technology devices to track your metabolic health, digital applications to help you find and learn about food and consumer products, and more. One product I use every day is the AquaTrue water filter. I recommend this to everyone because it gets everything out of your drinking water. Heavy metals, microbes, endocrine disruptors, microplastics, you name it. Other charcoal or gravity-based filters that a lot of people use don't get everything out, they just get some of the stuff out. So I really like the AquaTrue product. You can look for a link to my affiliate partners on my support page or directly in the episode description to this episode, or you can go to mindandmatter.substack.com to find it there.